Thanks, Tom. It's a pleasure to have a chance to present to the group. So you see the title of the talk, and it's embedded in work going on as part of a program project grant that is in the radiobiology program in the Cancer Center that involves myself, Joanne Sweezy, Patrick Sung, and Sally Rockwell from the radiobiology group, as well as Alan Sartorelli from developmental therapeutics. Some of this work started when I was at a Gordon conference more than 10 years ago, where people were talking about why are there so many mutations in cancer cells. And being associated with the radiobiology program, I started to think about the environment that cancer cells actually exist in, which is quite different from the normal environment we're used to in cell culture, because it's characterized by insufficient blood flow, leading to vascular insufficiency, hypoxia, low pH, fluctuating nutrient levels. And an example of imaging of hypoxic regions in tumors is shown here by staining with this compound EF5. But the hypothesis was, could this unique tissue environment cause altered DNA repair and replication, leading to genetic instability, and potentially influence cancer therapy? So we did a simple experiment at the time which tested this hypothesis, in which we grew cells that had a mutation reporter gene, so you could score it either in culture, or they were the type of cells that would form xenografts in nude mice as tumors. And consistently, the frequency of mutations that was higher in cells grown in vivo in tumors than grown in culture, suggesting that the in vivo microenvironment was somehow causing genetic instability. We subsequently took cells and put them in special chambers where we could control the atmosphere, and we showed that low oxygen levels also lead to an accumulation of mutation. This is all published, so I'm just showing you a cartoon diagram, but this was the fundamental observation. Then we went on to say, well, what's going on? And we spent a few years looking at increased DNA damage and things like that. And then taking a candidate approach, we initially identified that certain aspects of a DNA repair pathway, namely the DNA mismatch repair pathway, were specifically downregulated in hypoxic cancer cells. So as you may know, the DNA mismatch repair pathway recognizes base pair mismatches and corrects them, and has proteins with names like MLH1, PMS2, and MSH2. And actually, defects in this pathway are linked to hereditary colon cancer, the so-called Lynch syndrome, hereditary non-polyposis colon cancer to be specific, or HNPCC. And what you see here is a blot showing decreased levels of MLH1, one of these mismatch repair genes, in hypoxic versus normoxic cells. And just to point out something we'll mention a little bit later is, in addition to downregulation in hypoxia, the MLH1 gene specifically is silenced in sporadic cancers of multiple sites. And we believe this is linked to the observation here. To make a long story short, with some of the published data, we looked at the mechanistic basis for this, and we found that a series of transcription factors in the MYC pathway, MAX, MINT, MAD, etc., it's an alphabet soup of transcription factors, are specifically altered in hypoxia and do a dance on the MLH1 and MSH2 promoters, leading to specific downregulation in hypoxic cancer cells. Now, what does that mean? Well, with decreased mismatch repair, you do get genetic instability, and that we demonstrated that. It also reduces ATP usage, because this pathway is a heavy user of energy, and that may assist in adaptation to low oxygen and increased survival, but also can lead to altered therapy responses potentially because of a differential DNA repair function. And it turns out these proteins also have a dual role in signaling DNA damage leading to apoptosis, so that's another potential change. Now, we went on to say, okay, what else could be altered in hypoxia? And we did transcriptome profiling, and this is just a graph of the number of genes either upregulated on the right or downregulated on the left. And if you look at genes upregulated in hypoxia, you see a lot of metabolic genes that are associated with glycolysis or the angiogenesis, such as VEGF. But if you look on the other side, you'll see that there are some DNA repair genes that are downregulated, namely, and this was the surprise for us, the BRCA1 and RAD51 genes that are associated with homologous recombination or homology-dependent repair are seen to be substantially downregulated. We also see MLH1. Not all repair genes are downregulated. Some are, most actually are quite stable. So we followed up on the BRCA1 story, and I'll just remind you that BRCA1 plays a central role in regulating homology-dependent repair of double-strand breaks and stalled replication forks. So, as I'm sure everyone in this audience knows, BRCA1 is associated with familial breast cancer, and there's been a huge amount of interest in figuring out how it does so when it's absent. And it turns out it has a role in gene expression, it has a role in chromatin modification and ubiquitination, and it also has a role in regulating DNA repair, and it participates with BRCA2, which is also a gene linked with familial breast cancer, and RAD51 in mediating homology-dependent repair, and somehow cooperates with the Fanconi anemia-related genes, which form a multi-protein complex that also participates in this pathway, and more about that later. So we went on to study BRCA1 regulation, and you can see here on a northern blot and a western blot that BRCA1 is actually suppressed in hypoxic cells. You can see here 2 different cell lines, and also on the protein level. Interestingly, it's also phosphorylated, as you can see from this gel shift, even at the same time it's being downregulated. And that's a little bit of a story for another time. So we went on to analyze, well, why is this happening? And we looked at the promoter region, and we found these sites for transcription factors called E2Fs, which are a family of factors that regulate transcription, and we have some nice consensus sites. Interestingly, the promoter of BRCA1 is just 200 base pairs long. BRCA1 is expressed this way, and an unrelated gene, except for proximity, NBR2 is expressed that way. BRCA1 is suppressed in hypoxia, but NBR2 isn't. So that makes for an interesting question for people with desperate interest in promoter function. So we went on to look at the E2F family, and most of you know that the E2F factors are either repressors or activators, and they interact with the retinoblastoma protein and related pocket proteins P130 and P107 to form these repressive or activating complexes. And we used the technique of chromatin immune precipitation, which allows you to ask if a factor is sitting on a promoter. And what you can see here in normoxic cells on the BRCA1 promoter, E2F1 and E2F4 are present, but in hypoxia, E2F1, which is an activating factor, comes off, and E2F4 increases its occupancy, along with some of these pocket proteins I mentioned before, and that's graphically shown here. So this is kind of a mechanistic basis for this regulation of BRCA1 in response to hypoxia. And you can prove this genetically by taking a protein derived from the human papillomavirus, E7, which in its oncogenic role actually inactivates retinoblastoma, but as a genetic research tool inactivates these pocket proteins by binding to them. And you can block this whole pathway by just sticking this in the cells. And if you do so, the downregulation in normoxia versus hypoxia is lost. So that's a nice tool to prove the mechanistic pathway. And I introduce this here because I'm going to talk about this tool toward the end of the talk in relationship to the new class of drugs called PARP inhibitors. So again, to summarize a lot of work in the lab, we found that there's a complex constellation of regulation of the homology-dependent repair pathway in hypoxic cells. There's a signal transduction pathway that's still being worked out that I didn't show you data on, that involves the ATM protein CHEK2 and involves post-translational modification of proteins. There's also a gene regulation pathway in which we get repressive complexes formed that suppress this pathway. Now, why would the cell want to repress homology-dependent repair? Part of the reason, again, is this is a pathway that's a very heavy user of energy. And when cells are hypoxic, energy is at a premium. And basically, cells shut down sort of non-essential functions. And this is seen now in the concept of autophagy, where cells actually start to cannibalize their own macromolecules in order to survive stressful conditions. And it is a pathway commonly seen in cancers. In fact, people are trying to develop drugs to stimulate autophagy pathways. But in any case, this is probably related to a cell survival mechanism. But as a result, it also leads to genetic instability and altered response to therapy, as we'll talk about. So we alluded to hereditary breast cancer associated mostly with BRCA1 and BRCA2. Obviously, most breast cancer is sporadic, but what this slide, taken from a review article, indicates is a fair amount of sporadic breast cancers are associated with decreased expression. I think this is an old number. It's not that high of a frequency, but the point is that that's an important feature that's seen in sporadic cancers, is sort of acquired BRCA-ness, it's called. And I believe that Lindsay Harris gave a talk here recently and touched on this for breast cancer. So we started to think, well, maybe this repression that you see in hypoxia could be linked to the silencing of a number of genes that's seen in cancers. And in fact, we went about to test that. In collaboration with Mitch Turker, we got a construct which has the BRCA promoter driving the HPRT gene in one direction and APRT in another. And the importance of these funny genes, these are factors involved in the purine salvage pathway, hypoxanthine phosphoribosyltransferase transferase. And the important point of this gene is it's selectable. So when cells express this or not, depending on the drug you add to the medium, you can select for cells that have that functional or cells that have it non-functional. So we put this construct into a certain type of mouse cells that were deficient in these genes, so we could follow the function only of the genes that we introduced. We grew them in and out of hypoxia for 3 weeks, and then we selected in a compound 6-thioguanine in which only HPRT negative cells would survive. So that means we're looking for cells where this promoter has been shut off. Now you take them out of hypoxia, so cells were in hypoxia. Now they're in 6-thioguanine, and you see you selecting for colonies. Now this is out of millions of cells, so the frequency is low. But the point is, a lot of these cells now have, in a long-term way, downregulated or silenced the BRCA1 promoter. If you also add trichostatin A, which is a histone deacetylase inhibitor, it's not one clinically used, but it's a very potent one, it tends to reactivate genes, and you can prevent that silencing or reverse it. So the point is that you can select for cells that have undergone silencing due to hypoxic treatment. And in fact, Yuhan Luo in the lab is analyzing the promoter in this construct to try to see what are the histone modifications caused by hypoxia and linked with silencing. So it turns out, if you look at a lot of sporadic cancers, and I just did a quick literature search before this talk, and I have some of the information here, a number of DNA repair genes are silenced in sporadic cancers of multiple sites. BRCA1, MLH1, the Fanconi anemia protein, FANC-F, and also the Werner syndrome helicase, which is associated with premature aging, but also is associated with, when it's silenced, with these cancers. And here are some of the frequencies. For some of them, the frequency numbers aren't reliable because the denominator is not large. But the point is that silencing of multiple DNA repair genes is seen in a variety of cancers. So the question is, what does all this mean for cancer therapy? Well, it means that both in the cancers that come from familial syndromes, as well as those where the DNA repair genes are silenced or are downregulated in hypoxia, there's a potential for developing targeted therapies that exploit that repair deficiency. So what I want you to do that, I want to introduce the concept of synthetic lethality. And the idea here is condition A, which might be a DNA repair defect, is non-lethal. Condition B by itself is non-lethal. This could be a drug that is otherwise gentle on the cells, but when it's given to cells which already have condition A, the combination is lethal. And this was actually demonstrated in a very elegant way a couple of years ago by 2 groups that were targeting BRCA-deficient cells with poly-ADP ribose or PARP inhibitors. And this is taken from one of those papers that now has led to a number of compounds in clinical trials, that, including a recent report in the New England Journal this past summer that looked positive for BRCA-deficient breast cancer. But the point is that poly-ADP ribose polymerase plays a role in base excision repair or single-strand break repair, in which this little gap is fixed. Okay, so it's an enzyme that plays a role in that pathway. If that gap's not fixed and the cell goes through replication, you get a disrupted replication fork, which normally the cell can handle using the BRCA pathway of homology-dependent repair to use this template to fix that one. So if BRCA is deficient, it can't do that, and the cells die. And these are some survival curves of the BRCA deficient cells dying in PARP inhibitors and the BRCA proficient resistant. And actually, the differences are bigger than you think because these are on log scales. But the point is, there's a differential effect. It's a synthetic lethal effect of the PARP inhibitor. So we said, well, okay, base excision repair has other players. And getting together with Joanne Sweezy, whose lab is just next to ours, we decided, well, why not target, instead of PARP, polymerase beta, which is just downstream of PARP in this same pathway of base excision repair that's shown here. And it's actually the polymerase that fills in those gaps. So the idea would be the same. This gap couldn't be fixed. If you block Pol beta, then you're going to have the same problem with the BRCA defect. So we actually used a candidate approach, and we identified a compound. Actually, it's lithocholic acid, abbreviated LCA here. It's just shown here. It's actually a bile acid, so we're not proposing this as a drug, but it's a proof of principle. And it turns out it's a specific inhibitor of polymerase beta, and this is work done in the Sweezy lab, in which we can show the polymerase function of Pol beta is blocked by lithocholic acid, because if you add a nucleotide, you'll have a band up here, and LCA blocks that. Polymerase beta also has a lyase function, which means it cleaves a fragmented end to clean it up. And in fact, that lyase function, which is demonstrated by a lower band here, is lost. And then the DNA binding function of Pol beta itself is disrupted by lithocholic acid. So here's the binding without lithocholic, and these open circles show there's almost no binding in the presence of lithocholic acid. So it's a specific inhibitor of the key functions of polymerase beta. And in fact, if you reconstitute the whole base excision repair pathway, you also block repair as shown here. So then if you take BRCA deficient cells, you can show that they're sensitive to lithocholic acid. They're also sensitive to a factor temozolomide that engages base excision repair by causing a lot of methylation damage on purines. And that's shown in two different matched pairs of BRCA deficient cells shown here. But the interesting thing is when you combine those two, so you engage base excision repair and then you inhibit it, therefore creating a lot of those single strand breaks, you get a lot of synergism here with polymerase beta inhibitor and temozolomide as a proof of principle. And in fact, a similar study is being done has been reported using a PARP inhibitor and temozolomide. So it looks like this type of combination is very promising for BRCA deficiency. In the interest of time, I'm going to skip this slide. And this slide just shows that when you combine temozolomide and lithocholic acid to inhibit Pol beta in the setting of BRCA deficiency, you have persistent double-strand breaks, which are identified here by foci of a histone modification, gamma H2AX, that signals the presence of persistent double-strand breaks. So that's providing the mechanistic basis. So I'm going to have to move along a little bit faster, but let me just return to Fanconi anemia. I mentioned that this is an important player in the homologous recombination pathway. So we decided let's develop some drugs, or look for drugs that are synthetic lethal when that pathway is deficient, similar to PARP or similar to lithocholic acid. So instead of a candidate approach, we're taking a screening approach using the Yale Small Molecule Discovery Center. So we compared cells deficient or proficient in this recombination pathway. So this is related to the BRCA pathway. It's another player. And we have a matched pair of cells. And these are our hits. So actually right here, you can see, I think this one's the red one here. That's mitomycin C, which is a known agent that's toxic to Fanconi or BRCA deficient cells. And in fact, mitomycin was proposed for clinical trial in Britain for BRCA deficient cells based on this property. So we basically drew a line here and said, okay, these are all potential hits where there's differential effect on the deficient cells. And actually, the interesting thing that we're really excited to follow up on, I can't give you too much detail, is that several of these are known drugs that are used for non-malignant indications. So, you know, the pathway to using them in people would be very straightforward if the results can be validated. So stay tuned for more on that. And this is just some of the data showing the effect. This is the deficient cells, the cell killing, versus the proficient cells. So again, here is the synthetic lethality, some of the top hits. We also started screening for MLH1 deficient cells, and we have a nice compound that's more toxic to the deficient cells. And again, more on this later, but I think that this is an interesting approach. It's using some of the new facilities in the Cancer Center, and we're starting to identify compounds. I'll tell you, this is also a drug that's used on people, but not for cancer. So we'll see. Now, one thing I wanted to mention, there's also the concept of synthetic lethality in terms of radiation, but this has to do with fractionated radiotherapy. So this is a survival curve, and this is radiation dose and survival. So this curve here is for what we're all used to for oxic cells, that is, cells under normal conditions. Well, it's been known for decades that cells that are put in low oxygen and then irradiated in low oxygen are resistant. That's one of the problems with hypoxic cells. And actually, Jim Fisher, who's in the audience here, has worked a long time studying that problem. And so here's a PET scan showing hypoxia, and this is actually a tumor from dogs. But this is a PET scan showing this region a day after a fraction of radiation, where there's been reoxygenation of the area following radiation because cells have been killed off that were consuming oxygen, and now that area can be reoxygenated. So this happens with fractionated radiation, but what does that mean? So this is the hypoxic cells. But now, what you see here is these are cells that are hypoxic for a long time and irradiated in hypoxia. Well, they're a little more sensitive. Well, why is that? Because BRCA1 is down. Okay? But they still have the benefit of low oxygen reducing the impact of the radiation due to the chemical effect of oxygen. Well, here's the important one. So these are the cells that were hypoxic. BRCA1's been downregulated, but it takes a while to be re-expressed. So then when they're irradiated the next day and they've been reoxygenated, now irradiated in the presence of oxygen, they're the most sensitive. So in fact, this is in a way synthetic lethal because you've got a BRCA1 defect in reoxygenated cells, and they're the most sensitive to radiation. So it's one of the reasons probably that fractionated radiation is effective. Now, I don't have a lot more time, and I want to leave time for Brian, so I'm just going to mention that Denise Hegan in the lab was following up on the PARP inhibitor story, again a synthetic lethal story, and we said, well, probably hypoxic cells that are in which BRCA1 is down, they'll be sensitive to PARP inhibitors, and in fact, we found that. Here's normoxic and hypoxic cells. These are PARP inhibitors. That's fine. But it turns out that the story with PARP inhibitors, that normoxic cells there's no effect, is not completely true. Here's a normoxic cell. There's a little bit of an effect of the PARP inhibitor. But interestingly, when we use that E7 tool, it completely rescues any toxicity. So what's going on? Well, it turns out that if you put cells in PARP inhibitors for a day or two, you start seeing downregulation of BRCA1 itself. And you see this with numerous inhibitors and numerous cell types. You even see it if you use RNAi to knock down PARP. So, and in fact, it's reversed by E7, and the reason is because inhibition of PARP engages that same pathway that hypoxia does of downregulating BRCA1. So what does all that mean? Well, interestingly, it's been known for a number of years that PARP inhibitors sensitize cells to radiation. So you see here, this is PARP inhibitor treatment radiation dose. But if you block the downregulation of BRCA1 with that E7 tool, you reverse most of that radiosensitization. So this was an interesting story just to say sometimes the PARP inhibitor story, which was presumed to be only inhibiting base excision repair, it actually has multiple effects on repair pathways. So I think I'll stop there. And what we've talked about is that hypoxia downregulates and silences repair genes, and that this could lead not only in hypoxia but also in cancers with mutated or silenced genes to the design of targeted therapies, such as hitting Pol beta, base excision repair inhibitors, and also radiation effects, and that we have some small molecule screens underway. And these are some of the people that in the lab that have been doing the work. Beth Ross, Peterson Roth has done a lot of the screening. Greg Stahalik did some of the LCA work. And Eric Geiger did the work on the MLH1 in collaboration with Joanne. Thanks.